Welcome to semester 2 of the Human Biology content for year 11. The first lesson we're going to be looking at is DNA and what it is and how it's made. Now we're just going to start by looking at this image of a mother holding her baby, and then some years later that same baby being now a mother holding her baby, and it's really striking to see the similarities in these images, and it really gives us an understanding of how DNA is an instruction code that is inherited. Keywords for this lesson, hopefully they should be familiar to you from your junior school classes. There will be some extras, of course. First one is deoxyribonucleic acid, which of course is DNA. And then we're going to be looking at some new words that are histones and chromatin. Hopefully chromosomes, nitrogenous base, double helix, complementary will all be familiar to you. We will then talk about the type of bonding that occurs in DNA, which are hydrogen bonds and sugar phosphate bonds, also known as a covalent bond. And then we'll look at the 4 types of base in DNA, which are adenine, thymine, cytosine, and guanine. These are the learning objectives associated with today's lesson. I strongly advise that you read the relevant pages in your textbook before looking at this lesson to give you some background understanding. Okay, so we're going to start by looking at what DNA actually is, and you will need to complete notes on page 4 of your booklet as we go. First of all, DNA is found in the nucleus of eukaryotes, so that's eukaryotic cells. Humans have eukaryotic cells, if you remember, and that simply means that our cells have organelles which are encased in a membrane. Now the word polymer here means many, so poly means many, so you have an organic piece of information which is made up of many repeating units. And in DNA, the polymer is that twisted helix ladder that we traditionally see. And the monomer is the single nucleotide which you can see here and is enlarged in this diagram down here. Now, one nucleotide, which is in this box, is composed of 3 key things. Those are the nitrogenous bases. There's 4 different types of base. Here we are showing cytosine. We then have the deoxyribose sugar, which is that pentagon. And finally, we have a phosphate group, which is denoted by the letter P. Now this arrangement is really important, and part of the skill in this topic is that you may be asked to draw a nucleotide and label it with the relevant parts, or you may be asked to draw a short section of the polymer of DNA. So please make sure that you note where the bonds are found and that you are able to draw a simple diagram. So we're going to start by looking at those bases in a bit more detail. Now, as I've said, there's 4 different bases that make up DNA, and they bond together in a very particular way. Now, how they do this is called complementary base pairing. Adenine will always pair with thymine. It will never pair with anything else. So adenine will pair with thymine, or conversely, thymine will pair with adenine. Secondly, we have cytosine will only ever pair with guanine and vice versa. And you can see that in this simple cartoon diagram of DNA. We can see the sugar, the green circles are representing the phosphate, and then we have guanine and cytosine here, and then thymine bonding to adenine. Now, it is really important that you're able to spell these words correctly. You're no longer in junior school. It's not good enough to say A, T, C, and G. You must be able to spell these words. Please try your best to use the correct spelling. You won't be penalized for misspelling one letter or 2, but try and get it correct as far as you can. So now that we understand that DNA is made of these repeating nucleotides. We're going to look at how they actually bond together. Now, if we look at the backbone, nucleotides are held together by this strong covalent phosphate sugar backbone. And this makes the uprights of the DNA ladder, which you can see in the pink and the yellow. Now, this little circle here is showing you where that bond is. So without the circle here, we can see that the phosphate connects on to the pentagon sugar. And that is the bond that you see all the way up the uprights. And with this other side, we're going down the way, we can see the phosphate bonding onto the sugar at these points. Now, in between the uprights, we have the bases, and the bases are held together by weak hydrogen bonds. Now, these bonds are weaker, so they are denoted by this dotted line to show that they are weaker, and this forms the rungs of our DNA ladder. Now, once we have that bonding securely in place, the two strands of DNA will actually twist around each other to form a spiral known as the double helix. Now, it's really important when you're discussing and describing DNA that you use the words that I've highlighted in red. It demonstrates your understanding and that you know how to describe this correctly. So here we have a diagram again, just showing the non-twisted version of that DNA molecule, and here we can see what it looks like when it's all twisted together. Please make sure that you're able to draw a single nucleotide as is shown in this dotted oval, and that you're able to maybe draw 2 or 3 together bonded. So 3 on this side with a base, and 3 on the other side with their complementary base partner. Alright, so at this point we're just going to stop and do a little quiz. Please make sure that you are able to answer these questions. Pause the video here, write down your answers, and then we'll check the answers on the next couple of slides. Okay, hopefully you were able to answer those questions, and here are the answers. So first of all, when we're drawing a nucleotide, we can see the individual nucleotides here with the phosphate, the deoxyribose, sugar, and the base. If you wrote sugar here, that's fine, you'll still get the marks. Now, when you draw 2 nucleotides together, the best thing to do is to draw one on top of the other as shown here, and then just simply draw that line to show that strong covalent phosphate sugar bond. Number 3, the bases are adenine, thymine, cytosine, and guanine. And the base pairing rule for number 4 is adenine always pairs with thymine or conversely. And cytosine always pairs with guanine. Again, you can set that the other way around. The element present in all bases is, of course, nitrogen, hence they're called nitrogenous bases. And the bonds that hold the bases together, the complementary bases together, are hydrogen bonds. Hopefully you were successful in those questions. All right, bonus question here. Now that we know the complementary ways that the bases pair up, can you identify the complementary strand of this DNA sequence? So which one would be complementary? Hopefully you were able to identify that, and the answer, if you didn't get it, is B. So T or thymine always pairs with adenine, adenine pairs with thymine, cytosine with guanine, and so on. All right, now that we've understood the basic structure of DNA, now we're going to be looking at chromosomes, and that's what you can see in the background of these pictures. So here we're going to be looking at the structure of a chromosome. Now, we understand that DNA is a very long molecule, so in order to fit such a large amount of material in a very small space, it becomes tightly coiled up. So if you think about other structures that we looked at in the previous section, we've talked about, for example, the small intestine. That is 6 meters long and is tightly coiled up in a small space in your bowel. And within that, you have the villi and the microvilli, which increase the surface area to volume ratio. You have a similar thing happening here with DNA. So if you look at this cartoon here, this is our DNA molecule, and you can see that it's wrapped around these spheres. Now these spheres, there's 8 of them, and they're called histone proteins. So we have 4 on top and 4 underneath, and that together makes the histones. Now, when the DNA wraps itself around histones, we call it a nucleosome. So what you're seeing here is one nucleosome. And within DNA, there are many of these nucleosomes. And the reason why we have that is almost like a spool to wrap our DNA thread around to save space. Now, there are a lot of words that sound very similar here, so let's just go through them. So the next two we're going to be looking at are chromatin versus chromosomes. So chromatin is this almost like an unraveled ball of wool. So it's this tangled mass of DNA that isn't all neatly wound up. So in this image here, you can see chromatin, and this is when a cell is not dividing, it's not undergoing mitosis, so it doesn't arrange itself all neatly. However, in the cell next to it, which is about to undergo cell division, the chromatin winds itself up into these neat chromosomes so that it can organize itself better. So chromatin is this DNA that is this mass of tangled DNA, whereas chromosomes are the chromatin which has become supercoiled and condensed to form these structures that are easily usable, is a better word for it, I suppose, but they're easily manipulatable when we are about to have cell division. So for reference, you should know that humans, healthy normal humans, have 46 chromosomes, and each section of a chromosome codes for a gene. And we'll come back to talk about that later. So just to make sure that you understand the order of things, here's a little diagram to help you get your head around it. So we have DNA, which is a double helix composed of repeating nucleotides, and those nucleotides are the single basic structure of DNA. Now the nucleotides consist of a phosphate group, a deoxyribose sugar, and one of 4 nitrogenous bases. And you can see the bases forming the rungs of the ladder here, and the ribbon that you can see in blue represents the sugar phosphate covalent backbone. Now, this is a large piece of material, so in order to condense it and wrap it up into a smaller space, DNA wraps around a group of 8 histone proteins, and that section of DNA wrapped around those proteins forms nucleosomes. Now, nucleosomes, you can see here these little spools with DNA on them, coil up to form chromatin. And chromatin will eventually supercoil up, which is what you're seeing in this picture, to form chromosomes, which are what we see when a cell is about to divide. So here you can see a double chromosome. So use this information to help you complete page 5 of your booklet. So now that we talked about the DNA that's found inside the nucleus, we're going to be looking at another type of DNA that you have, and this is found in the mitochondria. Now previously when we talked about aerobic respiration, we touched on the fact that there is some DNA found in the mitochondria, and this alludes to our evolutionary past, which we'll talk about in year 12. But we're just going to compare those two right now. So nuclear DNA exists as chromosomes, and it's found inside the nucleus of eukaryotic human cells, and it codes for pretty much all of our key human genomes. So that's about 99% of all our genetic code, and it's inherited from both our parents. So that's our genetic maternal and paternal parents. Mitochondrial DNA, on the other hand, is slightly different. It exists as these circular discs compared to the chromosomes that we traditionally know about in nuclear DNA. It's only found inside the mitochondria, and it codes for about 1% of the human genome. Now, even though it's only 1%, it actually does some really important things. Now, it actually helps with cellular respiration and protein synthesis, which are two metabolic functions that are essential for life. Now, the interesting thing about mitochondrial DNA is that it's only inherited from the maternal parent, so that's the mother. You'll learn more about that next year. Now, if you're interested, there is a really good article from the BBC about mitochondrial disease and how science has managed to manipulate this to make a 3-parent child. Okay, so just to help you complete your notes on page 5 of your booklet, here is a summary of the differences between mitochondrial DNA and nuclear DNA. Now, the key differences that you can see are that the shape of it is different. We have a double helix in nuclear DNA and the circular rings in mitochondrial. And the functions, the wide and varying functions of nuclear DNA are to code for genes and proteins, whereas mitochondrial DNA codes for respiratory enzymes and a molecule called tRNA, which we'll talk about later. Now, because nuclear DNA is so large, it is wrapped around histone proteins and forms nucleosomes, whereas mitochondrial DNA does not, and it's just a single circular ring. All right, so that's the end of this section of the lesson. What I'd like to do is to stop here and complete pages 4 and 5 of your booklet, and to complete questions 1 to 3, 7 and 8 in your textbook on pages 246 and 247.